In 1974, geologist Jim Bowler made a discovery that would shake the scientific world to its core.
So that was a moment when Australian history changed.
A skeleton, male, carefully buried in the red earth of what's called the Lunette Dune. And here's what made it extraordinary. The body had been sprinkled with ochre. This wasn't a casual disposal of remains. This was a burial with intention. They called him Mungo Man. And within 3 decades, his DNA would become the most contested genetic evidence ever recovered from Australian soil. At roughly 42,000 years old, he became known as the oldest human skeleton on the Australian continent. Here was physical evidence of Australia's first people, preserved in extraordinary detail. The ochre burial suggested sophisticated cultural practices. But the most controversial question about this skeleton still hasn't been settled. If we're right about him, it rewrites how the first Australians arrived. To understand what makes Mungo Man so controversial, we need to go back to where he was found. The Lunette Dune, a crescent-shaped ridge on the eastern shore of what was once Lake Mungo, part of the vast Willandra Lake system. When water levels were high, tens of thousands of years ago, waves deposited sand and clay along the lakeshore. As the climate dried and the lake gradually disappeared, wind carved these deposits into distinctive dune formations. It's a time capsule, written in sediment and sand. Every layer represents a moment in history, a snapshot of life on the shores of an ancient lake. Radiocarbon dating placed Mungo Man's burial at approximately 42,000 years before present, the oldest human skeleton on the continent. It was a landmark discovery, pushing back the timeline of human habitation in Australia by thousands of years. But here's the twist. The oldest skeleton might not mark the earliest people. Regional finds, like remains and artifacts from nearby cow swamp, along with stone tools, ancient hearths, and other archaeological evidence, push signs of human presence in Australia back even further, at least 47,000 years, possibly more. So, while Mungo Man is the oldest skeleton we found, the human story in Australia likely stretches even earlier. There may have been thousands of years of human history before Mungo Man was laid to rest on that dune. This distinction matters. It tells us that what we found is just a fragment of a much longer story. Mungo Man isn't the beginning. He's a chapter in a book that started much earlier. Everything looked neat when he was first discovered. A date, a dune, an ochre burial. The archaeological picture seemed complete and tidy. Scientists had a skeleton, a burial context, and a timeline that fit with other evidence of early human migration into Australia. Then, the genetics crashed the party. In 2001, a team of researchers published a bombshell paper in the Proceedings of the National Academy of Sciences, or PNAS. They claimed that Mungo Man's mitochondrial DNA belonged to an extinct lineage of modern humans, a population that arrived in Australia before Aboriginal Australians. Let me repeat that, because it's a staggering claim, before Aboriginal Australians. The theory was dramatic, and would reshape our entire understanding of human migration. Multiple waves of migration into Australia. An earlier group of people, possibly replaced or absorbed later by subsequent arrivals. Maybe even traces of archaic hominins, ancient human relatives like Neanderthals or Denisovans, mixing into the population. If true, that would mean a lost population, possibly replaced later, and maybe even traces of archaic hominins in the Australian fossil record. It would mean that Aboriginal Australians weren't the first people on the continent, that there was an earlier, now extinct population whose story had been lost to time, that the human history of Australia was far more complex and layered than anyone had imagined. The researchers used PCR, polymerase chain reaction, a technique that amplifies tiny amounts of DNA to make them detectable. They worked with Mungo Man and another specimen from the region, Cow Swamp 8, abbreviated as KS8. Both skeletons were ancient. Both offered the tantalizing possibility of recovering genetic material from Australia's deep past. They reported mitochondrial sequences that fell outside modern Aboriginal diversity. The genetic markers didn't match anything seen in living Aboriginal populations, a distinct population, different from anyone alive today. The DNA seemed to tell a story of a separate lineage, a ghost population that had vanished from the genetic record. The scientific community was stunned. Some embraced the findings as evidence of complex migration patterns. Others were skeptical. The claims were extraordinary, and in science, extraordinary claims require extraordinary evidence. But extraordinary claims need extraordinary proof, and the proof collapsed the moment newer methods hit the same bones. Years later, a team at Griffith University in Queensland, led by geneticist David Lambert, decided to re-examine the claims with modern technology. Science had advanced significantly since 2001. DNA sequencing had become faster, more accurate, and more sensitive. New methods existed for handling degraded ancient DNA and distinguishing it from modern contamination. They used second-generation sequencing, a revolutionary technology that could process millions of DNA fragments simultaneously. They employed targeted capture techniques to fish out human DNA from the complex mixture of soil bacteria, fungi, and degraded material in ancient bone. And they used something called SPEX, single primer extension, specifically designed to handle the short, damaged DNA fragments characteristic of ancient samples while reducing the risk of contamination. The team took extraordinary precautions. They cleaned bone surfaces with sodium hypochlorite, essentially household bleach, to strip away modern contamination that might have accumulated over decades of handling and storage. DNA extraction happened in sealed, clean rooms with positive air pressure to prevent airborne contaminants from entering. Researchers wore full protective suits. Every step was documented, controlled, measured. Their goals were clear and methodical. First, confirm that the DNA they recovered was human and not from bacteria or other organisms. Second, assess whether it was genuinely ancient by looking for characteristic damage patterns. And third, characterize the complete mitochondrial genome to see where it fit in the human family tree. The question they were really asking was simple but profound. So, who really left those European genetic fingerprints on Mungo Man? The answer wasn't in the ground, it was in the lab. The results were stunning, but not in the way the 2001 paper had suggested. Mungo Man's extracts carried 5 different European mitochondrial haplogroups. 5. Not one ancient signal from a lost population, but multiple modern European lineages all mixed together in the sample. When you see multiple European haplogroups in a single ancient sample, that's not a biological reality. It's a massive red flag for contamination. Ancient DNA doesn't work like that. One person has one mitochondrial lineage inherited from their mother. You can't have 5. This was contamination, pure and simple. Modern DNA from multiple sources had infiltrated the ancient sample. And Calswamp 8, the other specimen from the 2001 study, it yielded no authentic Aboriginal DNA at all, 0. The original sequences that had been reported couldn't be replicated with modern methods and controls. They simply disappeared when proper protocols were applied. Multiple European haplogroups flag modern contamination, not an ancient European in Australia. The extinct lineage wasn't ancient at all, it was modern, and one clue pointed to a very human source, not prehistoric, but present day. A specific possibility emerged during the reinvestigation. One of the 2001 researchers' DNA, specifically Gregory J. Adcock, matched a European haplogroup seen in the original extracts. The genetic signature that had been interpreted as ancient and exotic might have come from someone in the lab who handled the bones. To be sure, to have absolute confidence, they needed a cleaner test case, and it delivered not one genome, but 2. Enter WLH4, another skeleton from the Willandra Lakes region. But WLH4 was different. It was a late Holocene burial, only 300 to 500 years old. Much more recent, less mineralized, better preserved. The bone quality was superior, the DNA less degraded. It was the perfect control specimen. WLH4 produced 2 complete mitochondrial genomes, 2 distinct, full-length sequences. One was labeled S2, the other was labeled V3C. S2 showed all the hallmarks of ancient DNA. The molecular signature of time was written all over it. Cytosine deamination, a characteristic form of chemical damage that accumulates in DNA over centuries. Damage patterns concentrated at fragment ends, exactly where you'd expect environmental degradation to attack first. Short fragment lengths, typical of degraded ancient material. Every marker pointed to authenticity, and it clustered perfectly within Aboriginal diversity. S2 belonged to haplogroup S, one of several mitochondrial lineages found in indigenous Australian populations, along with haplogroups O, P, and M. These are the genetic signatures of Aboriginal Australians, distinct from populations elsewhere in the world, and reflecting tens of thousands of years of isolation and continuity on the Australian continent. V3C, on the other hand, told a completely different story. It was a modern European contaminant, clean, undamaged, no signs of ancient degradation whatsoever. It had all the characteristics of fresh, contemporary DNA, and it was sitting right there in the same sample, right beside the ancient sequence. This was the smoking gun. One sample, two genomes. One ancient, one modern. It demonstrated beyond doubt that ancient and modern DNA could coexist in the same bone extract, and that only proper damage pattern analysis could tell them apart. Ancient damage marks S2 as authentic Aboriginal ancestry. V3C exposes modern contamination sitting beside it. The contamination didn't erase the ancient signal, it just obscured it, confused it, made it nearly impossible to interpret without modern sequencing methods and rigorous quality controls. With clean ancient DNA finally in hand, the team could ask the question that really mattered. So, how deep do these roots go? Using Bayesian phylogenetic analysis, a statistical method that estimates evolutionary relationships and divergence times, they traced S2's place in the human family tree. They estimated the most recent common ancestor of S2, the point where this lineage split from its closest relatives, at around 28,500 years ago. The broader S2 lineage, encompassing related subgroups within haplogroup S, dated back even further, approximately 35,500 years. These aren't just numbers, they're chapters in a continuous story. They represent real people, real lineages, passing mitochondrial DNA from mother to child across hundreds of generations. These dates fit a long, continuous Aboriginal presence, not a vanished pre-Aboriginal population. They align with archaeological evidence. They align with linguistic diversity. They align with cultural traditions that speak of deep connections to country, stretching back to the dreaming, to the time of creation itself. The genetic evidence and the cultural evidence tell the same story: continuity. An unbroken thread connecting living Aboriginal Australians to the people who walked the shores of Lake Mungo 42,000 years ago. That dramatic extinct lineage claim, it didn't survive modern testing, but the science isn't the only record. There's another story here, one that existed long before DNA sequencing, one that will persist long after the last genetic analysis is complete. When Mungo Man was buried 42,000 years ago, his body was carefully positioned, laid to rest with intention, sprinkled with ochre, red earth, sacred earth. Ochre isn't just pigment. In Aboriginal culture, it's deeply symbolic. It represents life, blood, earth, connection to country. Its use in burial suggests spiritual beliefs, a worldview that saw death not as an ending, but as a transition. This wasn't just a body, it was a message. When you bury someone with ochre, you're not just putting them in the ground, you're sending them onward, you're honouring them, connecting them to country, to ancestors, to something bigger than any one life. The ritual speaks to a sophisticated understanding of death, spirituality, and community obligation. Ochre has been used across Australia for tens of thousands of years, in ceremony, in art, in marking significant life events. Ancient rock art sites show ochre paintings dating back 40,000 years or more. Ceremonial use continues in Aboriginal communities today. The red pigment is a thread connecting past and present, ancient practice and living culture. The care, the ochre, the ritual, they speak to a sophisticated cultural world tens of thousands of years old. This isn't primitive or simple. This is a fully realized human culture with complex beliefs, artistic expression, and social structures. The burial of Mungo Man is evidence of all of that. Frozen in time, preserved in sand and pigment. But that ceremony, that careful burial, was about to spark one of the most heated controversies in Australian archaeology. Along with Lady Mungo, known as LM1, another ancient skeleton from the same region, he was reburied at an undisclosed location near the lake. The ceremony was private, respectful, attended by community members, elders, and a small number of scientists who had worked with the remains. It was done in accordance with community wishes. The traditional owners of the land, the people whose ancestors walked those dunes tens of thousands of years ago, wanted their ancestral remains returned to country, wanted them laid to rest properly, permanently, with dignity. Honouring community wishes closed one chapter, but did it close the door on future discoveries? Reburial presents a genuine tension in science and ethics. We have technologies now, nuclear DNA sequencing, ancient proteomics, isotopic analysis, techniques that weren't available even 5 years ago. And they'll keep improving. Technologies we can't even imagine yet will be developed in coming decades. Some scientists worry we've lost the chance to learn more, that information locked in those bones is now inaccessible forever. But that's the wrong framing. Our ancestors aren't specimens. They're not objects to be studied forever, curated in museum drawers, poked and prodded with every new technology that comes along. They deserve rest, they deserve dignity. And the living descendants, the communities who maintain cultural and spiritual connections to those ancestors, they have the right to make decisions about their treatment. Science has given us answers. Good answers. We know now that Aboriginal Australians have been on the continent for at least 47,000 years. We know that genetic continuity stretches back at least 35,000 years. We know that the extinct lineage claim was contamination, not biology. We know that Mungo Man's burial represents sophisticated cultural practices 42,000 years ago. Those answers matter. They're significant contributions to human knowledge. Lake Mungo stands as a testament to that story. A place where science and culture intersect, where ancient bones tell modern stories, where the past is never truly past but always present, always relevant, always part of the living landscape and the living community. The mysteries may be solved, but the wonder remains.